Okay, so we finished the first two rules. Let's look at the rule number 3. Rule number 3, some chemical reactions can can proceed in either direction or two direction. So sometimes you see one arrow. One arrow means they just go from left to the right. But this one you see two. Two means they can go in this direction, they can go back. When you take AP2, you will learn, and also AP1, this is a very common equation you see in the body. CO2, that's the gas molecule your cells produce. And when your cells produce CO2, and it dissolves in blood, blood has water, especially the plasma part, water, and it becomes carbonic acid, H2CO3. So also this is when you drink soda, soda have a lot of bubble. That's they push CO2 into the sugar water. That's why soda is acidic, H2CO3. So they can go in both direction. When you have too much H2CO3, they can go back, become CO2 and water. That's why when you drink soda, you see a lot of bubble. These bubbles are CO2. So when you drink too much, too quickly, you drink soda too quickly, you burp. That's CO2. So they can go in either direction. And that's another one. So these equations are double arrow. So you can go to left, go to the right. And also rule number 4, some chemical process require enzymes. We will talk about the enzyme in detail in the next few slides. Enzyme is a protein in your body. Its function is to speed up the process. So you have a lot of digestive enzymes in your body, and they provide the space for the chemical interaction to happen. So you have a lot of digestive enzymes in your digestive system to help you to speed up the digestion. It's a chemical process. So enzyme increases speed. And if you don't have enzyme, it can still happen. This chemical process can still happen. It's just much slower. So in your body, especially you take this class to take AP1, to take AP2, you will see a lot of chemical response. In your body, you have enzyme to help. Most enzymes, they end up with the ASE. So you see the one with ASE, you know it's enzyme, like sucrase. Sucrase is the enzyme help you to digest sucrose. And lactase is the enzyme help you digest lactose. Lactose is a sugar you find in milk. Some people don't have enough of this enzyme. And what happens is when they drink too much milk, their body could not digest it. Okay, and enzyme usually will put them above, superior to this arrow. So like these chemical reactions on the left, you know there's the original molecules. On the right, you know there's a product. Okay, and you see the arrow, it go in this direction, and you see this above the arrow, you know that's the enzyme to do this activity. And sometimes they can go back and forth. Okay, again, you know this is the enzyme for this chemical process to happen. So let's quickly look how does the enzyme work. So enzyme help the chemical process to happen, but the chemical process does not change. The original reactant, we call this reactant, and the final product, we call these products, won't change. The enzymes to help the process. It's like they provide the space, okay, and this original molecule substrate, they'll bind with the enzyme, and with the enzyme's help, they help to break down the bond, and now they have two products. So the reactant and the product won't change. And there's another way to explain the enzymes. Don't be scared by this picture. This picture tells you originally this reactant, we use reactant to describe the molecules. They are going to participate in a chemical response. And the product, that's the final molecules. To turn the reactant into the product, that you have an energy gap to go through. So when you take the chemistry lab, you find a lot of times when you do the experiment, you use temperature, you increase temperature. You use fire, you increase the temperature for the chemical process to happen. You have an energy gap you need to go through. But in your body, you cannot burn a fire, you're gonna burn a hole. So we use digestive enzymes to help the process to happen. So this enzyme is job is to lower the energy required. So if you don't have the enzyme, you have a big gap to go through. With the enzyme, you have a much smaller gap to go through. So it's much easier for this to happen. This enzymes work. So they just lower the energy required for the chemical process to happen. And if you're interested, you can look at this video. It will show you, you have a picture, you have a video to show you how the enzyme work. So enzyme just provide the space for the reactant to come, and then for the chemical process to happen much easier, you have the product. That's the enzyme's function. And the reactants also called substrate, just different way to say the same thing. Okay, so enzymes, they have active site, they have a binding site. When we talk about proteins, we will go into more detail. Every proteins have their correct 3D structure for the chemical process to happen. And enzymes, they have a binding site, we call it active site, is specifically for the substrate to come, you'll bind with it. So each enzyme only fit for one substrate. It's like a key matching system. So you find your body have different digestive enzymes, like the carbohydrates. That's the enzyme you find in your small intestines. It only digests carbohydrate. And lipids is the enzyme digest lipid. You won't digest carbohydrate. So, and peptidase, that's the enzyme digest protein. So each one's only digest one molecules because they're very specific active site only for that substrate. So there's another picture show you the enzymes. They have specific binding site for the substrate, also called a reactant. And after they bind chemical interactions, they'll be released. That's the product. And the enzyme is available again for another substrate. You find enzymes, you just reuse them again and again because they never participate in the chemical reaction. They just provide the space for them to happen. So you have the bile. When you learn AP2, you will learn the bile. And bile is stored in gallbladder. It's actually released from your liver cells, but it's stored in gallbladder. And we reuse the bile. That's why we need to have gallbladder. Its function is to digest lipid, and we can reuse them every time you eat oil, and the bile will be released to digest the lipid. And after they've been done, they just go back to the gallbladder to be stored. So some people, they need to remove their gallbladder, and they found they could not eat the food with too much oil. So enzyme can be reused. Okay, so factors affect the enzyme activity, several factors. The first one is temperature. So enzymes, because it's a protein, so it has specific temperature, it will respond well. If the temperature is too high, too low, it will decrease its activity. So it has a perfect temperature, like the enzymes work in the body, most of them like your body temperature. And when you increase the temperature, the enzymes can move faster. So they can speed up the speed for the chemical process to happen, but only to a certain extent. Because when the temperature is too high, when you start to increase temperature, yes, it will speed up the process. But when the temperature become higher and higher, eventually it will destroy the structure of the protein. Eventually, the result is, okay, the protein won't work. Uh, that's why, okay, the speed increase and then it start to decrease. So when the temperature is too high, you can actually denature the protein. So this word denature means change the protein's 3D structure, so it won't work. And a good example is this. When we cook the food, the temperature, turn the egg into a fried egg. So it's still egg, still egg. So when you eat the fried egg, you still have all the nutrition you get from a raw egg. And we fry that, we find it totally changes structure. And it's irreversible, means it could not go back. And also it helped to destroy the bacteria inside if you have bacteria. That's why we cook the food, because bacteria are also proteins. So it helps you to denature the bacteria proteins. So it destroys the bacteria. And that's why we cook the food. Okay, let's take a short break.